Welcome back, ladies and gentlemen. In this video lecture, we're going to be looking over more inheritance patterns, in particular, extrachromosomal inheritance. We have the agenda lined up, a review of nuclear genetic inheritance patterns. Then we're going to look at, very briefly, the theory of endosymbiosis. And then we'll focus in on 4 aspects of this particular video lecture. The inheritance pattern for mitochondria, followed by the inheritance pattern for chloroplasts. And then we'll look at epigenetics. In this particular case, we're going to be looking at imprinting. And then finally, we'll conclude today by looking at maternal effect genes. Let's go straight into the review. We should know that with a single gene, say big A, small a cross between 2 heterozygotes produces a particular ratio, a pattern. So if you take 2 parents that are heterozygous and then you cross them together, then in the F1, you should get a ratio of 3:1 for the phenotype pertaining to the dominant versus the recessive. So 75% of the offspring will be phenotypically resembling the big A, and then 25% will phenotypically resemble the small a. So that's a single gene. Then, as Mendel did, he moved on to looking at 2 genes at the same time, a dihybrid cross. So that's a double gene, and we represent that by picking 2 letters out of the alphabet. So gene A and gene B. And we study both genes at the same time. So in a double heterozygous mating, we have 1 parent as a double heterozygote, and then we breed that parent with another heterozygote. And this time in the F1, we will have a pattern, and that pattern will be 9 to 3 to 3 to 1. And that's a phenotypic pattern too. So you should be familiar with those already. On the next slide, let's look at some departures that can be caused by the environment or by using various combinations of alternative chromosomes. So our favorite is X-linked traits. So this will be color blindness in humans, for instance. The gene itself resides on the X chromosome. If it's Y-linked, that could also be considered here. What you find in those cases is that you get a departure from the 3:1 ratio representing all offspring. And in this case, you get some kind of sex-related disparity. So there'd be more males with the condition than females. And you should be in a position to calculate that at this time. There's other instances too. You can get a ratio of 2:1, not 3:1 sometimes, when you're looking at lethal genes, where a particular combination of alleles causes the death of that individual, therefore reducing the number of progeny that are available in their classes. So these are all indications of departures from simple Mendelian inheritance to more complicated versions. We can also apply the same logic to a dihybrid cross. So you can see the total number here adds up to 16. So those are the 16 boxes that we see in a 4x4 dihybrid cross Punnett square. So if you get deviations, as we saw in the previous video, of 9 to say 7, that indicates that there's an interaction going on in some complicated fashion between the 2 genes that you're considering. Sometimes it's environmental, but most of the time, 1 gene is impacting the other's behavior. And there's a second example that I want to show you quickly. You can get a ratio of 15:1 when you had gene duplication. And that, again, is a departure from your 9 to 3 to 3 to 1. So there's many examples of this, and you should be familiar with those. So there's your groundwork. Let's now move on and look at the rules under which those groundworks should be effective. So the first thing it says is that meiosis is the type of cell division That's very important here. And all of these rules can be applied to some aspect of meiosis. So the first one says alleles segregate during gamete formation. So this is one of Mendel's laws. If you start with a heterozygous parent, what this means is that during the process of gamete formation, where meiosis is instigated, the big A allele will be separated, segregated, into a different cell than the small a. So big A goes into one type of gamete, and small a will then move into a different type of gamete. The same thing would apply with eggs. So you have segregation. The second rule says we have to assume that these alleles are stable. So a big A does not change into a small a. A small a does not change into a big A. And that also applies to big A not changing into something else. So we have to assume that. Even though it happens, and there's a very small chance that it can, we have to assume that it's not happening. Number three, that the phenotype of the F1, the offspring, is an interaction of the biochemical interplay between the different alleles that are present inside the cells of that individual. So if you have a heterozygous condition, then the interplay between these two alleles should be able to be explained at the molecular and biochemical levels. And then lastly, we go back to Mendel with independent assortment. So according to him, when you have two or more genes, each gene has no impact on the migration of the other gene into the gametes. So let's put that in a way that we can recognize. Suppose we have a heterozygous condition for the A gene, and we also have a heterozygous condition for the B gene. So when this individual here makes gametes, the gametes will contain at least one A, and let's assume it's the big A, then there's nothing to prevent either B allele from joining that particular gamete. So you can get two types of gametes. You can get big A, big B, big A, small b. And the reciprocal is also feasible at the same particular percentages. So you can get situations like that. So if it's a male, you get 4 types of sperm. If it's a female, you get 4 types of egg. So that's independent assortment. There's no linkage, if I can use that word, between the two genes. So there's your foundation. And you mustn't forget this, because this helps you remember and apply everything you learned previously. What you'll see now is that there's a departure from those rules. At least one of those rules will be violated by the systems that we study across the next 4 examples. Before we get there, we have to understand to a general degree what endosymbiosis is. And the theory of endosymbiosis simply says that we have evidence to demonstrate that this process occurred in the past during the history of life on planet Earth. So where do we start with this type of analysis? Let's keep it very superficial. And if you need to know more, there's plenty of videos on the internet. So the Earth is about 4.6 billion years old. So 4.6 billion years ago, the Earth was formed, okay? As well as the solar system. The surprising thing is, it didn't take long in respect for life to begin. So people reckon about 3.7 billion years ago, the first life forms took hold on the planet. So let's represent that as a cell, okay? And these cells were prokaryotic in nature. So they had no nucleus and they were relatively simple cells. And as time passed, these cells broke up into multiple branches through speciation. And as time passed, different cells acquired different abilities. The next major event with respect to endosymbiosis is about 1.5 billion years ago. And at that time, we had the prokaryotic cells, existing on the planet. And these cells were now able to perform complex biochemistry. At the same time, another branch of these cells developed into larger cells, and they began to form a nucleus. So these were the first eukaryotic cells on the planet. And some of these cells started feeding on the smaller cells. So we find that as time passed, some of these smaller cells were found inside the larger cells. So some prokaryotic cells were being consumed, probably by the eukaryotic cells for food. And there was a mutation. And in this cell here, that process came to a standstill. And the internalized prokaryote was not digested for food, but instead began to live inside. And these internal bacterial type cells eventually evolved into present-day mitochondria. And there's a bunch of evidence to suggest that's the case, biochemical and morphological. As time passed, these cells then evolved, and those that didn't have the internalized prokaryote, they had a disadvantage, because what the internalized prokaryote allowed the big cell to do was to maximize energy production from food molecules. So these cells, they couldn't break down their food molecules completely, so type of fermentation, whereas these cells here were able to completely break down food into carbon dioxide and water. So these cells continued, and these cells became extinct. But this led to the formation of mitochondria in all eukaryotic cells. The next story of endosymbiosis, part 2, has to do with chloroplasts. So let's continue from the previous slide. So we have prokaryotic cells on the planet, and we have these new larger eukaryotic cells that contain inside them evolving bacteria that will become present-day mitochondria. So the process continued, and of course, evolution doesn't stop. And these cells broke up into multiple branches, and so did these cells. They broke up into multiple branches too. Okay. So these ones still had the evolving mitochondria, and these ones were cousins, and they also had evolving mitochondria. And by now, these cells began to photosynthesize. Some of them were still heterotrophic. That means these cells here, they fed on complex chemistry, and these ones were able to capture sunlight and use that for combining carbon dioxide with water to produce their own food through photosynthesis. And we believe that a second endosymbiosis event took place where these types of bacteria were internalized by one branch of the eukaryotes. So we can use a different color there, if I may. Let me use green. So these are green photosynthetic cells. And as this continued, these cells were then internalized. So there they are. Okay. Not all of them, but some of them. And this branch continued unaffected. So the theory goes like this. This particular branch became today's fungi and animals, eukaryotic kingdom of fungi and the eukaryotic kingdom of animals. And then, of course, many of the protists are also of this type. But this branch, the one that had the internal chloroplasts, they became present-day plants and some of the photosynthetic protists, algae. So thus, present-day eukaryotic cells fall into two classes. Those that just have mitochondria, and those that have mitochondria and chloroplasts. I'm going to move on to talk about extranuclear inheritance with respect to mitochondria. So you see that I have 5 bullet points, and these 5 bullet points are just to give you a checklist of some of the characteristics of a mitochondrion. Okay? So the genome size of mitochondrion, that is relatively small. So the human one is about 17,000 base pairs. So considering that our nuclear DNA is about 3.2 billion, this is tiny, okay? And these mitochondria are roughly within the same ballpark across all the eukaryotes. Now, the chromosome type is circular. And we only find circular mitochondrial DNA in eukaryotes. This is further evidence that their evolutionary history is related more to prokaryotes than it is to eukaryotes. Now, the number of these genomes that you find inside a single cell varies across the eukaryotes. For instance, in mammals, you can find between 20 and 50 copies of the genome inside a single cell. So there's a big range of possibilities. So in mice, I think you have 2 or 3 hotspots within the mitochondria. So let's draw a mitochondrion. And it has an outer membrane. Then it also has an inner membrane. And let me just stop here and tell you, the outer membrane looks like eukaryotic plasma membrane. And the inner membrane has a chemical signature of a prokaryotic membrane. So let me demonstrate quickly how this works. If this was an ancient prokaryotic cell that became today's mitochondrion, it would have had its own membrane that you can see in white. And a larger cell would have consumed this by wrapping its plasma membrane and then internalizing it. Endocytosis is the process. Now, if you take a snapshot of this, you will see that it's surrounded by 2 membranes. The white one has a signature of a prokaryote cell, and the outer one has a signature of a plasma membrane of a eukaryotic cell. So that difference in chemical composition is one line of proof that the mitochondrion came from an endosymbiotic event in the past. The figure here can be used to relate to the information on the far side. So we have a circle of DNA inside the matrix of the mitochondria. In fact, what you do find is that there's 3 or 4, maybe 5 copies of this chromosome all stacked together into a blob called a nucleoid. And instead of having just one nucleoid, you could have another stack of chromosomes in another region of the same mitochondrion. And in many species, like mice, you may find, on average, between 1 and 3 of these. So if each one has 5 or so, or 6 chromosomes, then you have 6 chromosomes here, 5 or 6 here, and 6 here, and then you end up with about 18 to 20 per cell. But that's assuming that each cell has 1 mitochondrion. Metabolically active cells, like your muscle cells, can have hundreds of mitochondria per cell. So the number of mitochondria in a cell can vary from a few to hundreds. So then you have to multiply this number by the number of mitochondria inside that cell. Okay? How many genes are there in human mitochondrion genomes? And the answer is about 13 for coding for protein. So they make 13 different types of protein, all involved in oxidative phosphorylation, i.e. the complete breakdown of food. They also have ribosomal RNA genes for making their own ribosomes. And then they have tRNA genes, transfer RNA genes, that are useful for transferring amino acids to these internal ribosomes. So talking about the ribosomes that these mitochondria have are of the same order and same chemical composition as prokaryotic ribosomes. The ribosomes that you find in the cytosol of eukaryotic cells are larger, and they're made up of a different combination of ribosomal RNA, and they work with their own sets of tRNA. So what we're going to focus on next is how in mammals the pattern of inheritance of mitochondria take place. Keep in mind that different branches of eukaryotes, they do it in different fashions. We're just going to focus on humans and mammals. So suppose we have a father, and he has his gamete, the sperm. The sperm just has enough room to carry the nuclear genes, the genome, the nuclear genome, right? It doesn't have any space to carry mitochondrial genomes. Even though the tail of the sperm has lots of mitochondria, none of them make it into the fertilized egg. Whereas in the female, the egg, which is massive by comparison, has its own chromosomes inside the nucleus, and it carries a large number of mitochondria. And those mitochondria have their own chromosomes. So upon fertilization, the sperm don't deliver any mitochondria, and the egg has contributed all of them to the next generation. So the F1 will be produced here upon fertilization. So this will be the zygote. Now, occasionally, there was a court case in England where the paternity of a child was challenged, and there was an anomaly, and they were having to explain, the scientists, how the father's mitochondrial DNA ended up inside the baby. And we now know from research that in many mammals, there's a leakage of the mitochondria from the sperm into the egg on very few occasions, maybe a couple of times every 100,000 fertilizations. So it can happen, but it doesn't normally happen. So please be aware of that. So the consequence of this is very simple. We have mother to offspring inheritance, right? And then the F2 will all get their mitochondria from their mothers from the F1. So with humans, we can trace back mitochondrial DNA through our lineage to a particular female that lived about 200,000 years ago. And we're all related to her in some abstract way. So regardless, as far as we're concerned in this course, mitochondrial DNA comes from our mothers, and our mother got it from her mother, and it's never passed from the father to the next generation. So that's the inheritance pattern that you should be aware of. Let's turn our attention to chloroplasts. Slightly more complicated, but still very similar. So the genome size of the average chloroplasts is between 200,000. Some of them are 100,000, but about 1,500 to about 300,000, right? So let's put 100,000 just to be sure that we capture that. So the range is quite large, and these are many times bigger than the genomes contained within mitochondria. So the type of chromosome they have is circular because of the endosymbiosis hypothesis. The number per cell, again, it depends on how many there are inside each chloroplast and how many chloroplasts there are per cell. But that number can be hundreds, and in some cases, thousands. And how many genes do they carry? Well, in this case, they carry 100 to 200 genes. And these genes, once again, some of them are making proteins for the organelle, others are making tRNA and ribosomal RNA for exactly the same reasons we saw in the case of the mitochondria. Okay, just to finish off the story here, we know that bacterial cells or prokaryotes, they divide by binary fission. That is, that a cell gets larger and larger, and then it copies its DNA and then the DNA is separated into two halves, and then the cell just splits down the middle. So that's binary fission. Well, chloroplasts also reproduce in the same fashion inside plant cells. And guess what? So do mitochondria inside plant cells, and mitochondria inside animal cells. So this process of cell division is common to prokaryotes, chloroplasts, and mitochondria. Another line of evidence to suggest that mitochondria and chloroplasts, were once prokaryotes. The last thing is the way in which these organelles are inherited. So unlike chromosomes, which are sorted so that one daughter cell gets half and the other daughter cell gets half, but they're sorted evenly so that one of each chromosome 1 is separated. When it comes to these organelles, it's all about numbers and where they happen to be by chance within the cytoplasm of a cell. So here's a cell about to divide, a plant cell, for instance, and we get nuclear division through mitosis and meiosis, but the mitochondria or the chloroplasts will just be hanging around inside the cell. And depending on where they are physically, when the cell begins to form a cell wall, then you can see that these ones will be trapped on the left-hand side and these ones will be trapped on the right-hand side. So it's just a numbers game. And sometimes you have far more in one cell than the other. The final thing is that you can have mutations in the genomes of these organelles. So we go back to mitochondria. You can now see it's possible that you could have, within all these different genomes, mistakes and changes. So a single mother cell, like this one here, an egg cell, could contain many varieties in terms of its gene and alleles and mutants, as well as wild type, many forms of these genes, all within the various combinations. So we call that situation heterogamy, right? Heterogamy. That means they're not all the same, they're different. And if that's the case, that can then give the cell a varied phenotype. The same thing with chloroplasts. Because there's so many genomes per organelle, and there's so many organelles per cell, you can have variation amongst these genomes. And that complicates genetic understanding of how inheritance takes place. Regardless, there are about 200 different conditions in human beings that are related back to these few genes that are present inside our mitochondria. Why? Because these are metabolic conditions and they can interfere with many pathways of energy utilization. And the same thing with chloroplasts, right? Chloroplasts are used for capturing sunlight and then converting that into food molecules. So anything that interferes in terms of their mutations can disrupt the biochemistry of the entire cell. So let's look at the last point here now. On the next slide, we're going to look at inheritance patterns of mitochondria in one species of organism. We're going to go with the geneticist's favorite plant, the 4 o'clock plant, and we're going to look at inheritance of that. And we're going to focus in on the color of the chloroplasts. So chloroplasts are present in 2 different colors, right? So this is a plant cell, there's the nucleus, and inside the cytoplasm, we have, let's say, a couple of different types of chloroplasts. So these ones here, well, I should use the right color. So these ones here are green. And that's the wild type condition. So there's a gene that allows the chloroplast to construct pigments for capturing sunlight, and they're wild type. And then also in the 4 o'clock plant, there's a mutation. And that mutation causes the chloroplasts to not do an efficient job in making chlorophyll. So these ones are slightly green, but geneticists call them white because they're more white than they are green. So please remember that, right? As we go through with this example. So depending on which chloroplast is present in which cell, then that cell has the potential of being a different color. So let's look at that situation. Suppose we have cells that contain just the green chloroplasts. So those cells will be green, right? The chloroplast inside those cells will make that tissue appear green from the outside. We can have other regions of the plant that only have white. This is the nucleus, by the way. They only have white chloroplasts. So that tissue in the plant will appear white. And the last situation is just like this one here, in that you can have both green and white chloroplasts inside the single cell. And that also would make the cell appear green. So where does this word come from? Variegated. That applies when you have a single plant on which the leaves are a mixture of green and white. So you can have a large plant, 4 o'clock plant, where some leaves are green, some leaves are white, and some leaves are a combination of patchwork, like a cow, a black and white cow. So that leaf has a part green patch and a white patch. So if you want to see an example of that, you should be able to see one on the internet. So what we have to explain to you is how this inheritance pattern works. The secret lies in one answer, and the answer is this. The mother, the egg, defines which chloroplasts are passed on to the offspring. The pollen doesn't have room for chloroplasts. It only passes in nuclear DNA to the egg. So just like in the mammal. So if that's the case, we have to explain all the permutations under which we may find this inheritance. So let's do that. Let's pick a green color. Let's choose 2 parents, right? 2 plant parents. The first one is a male, and say all he has is green leaves, right? And then that is bred with another plant that has green leaves. And you can see here, the mother will pass in the normal chloroplasts, and all the babies will have green leaves. It doesn't matter what the father is, because the father does not pass anything in terms of chloroplast DNA into the offspring. So this is a maternal inheritance only. Okay, so let's change the situation slightly, and let's change the identity of the father just to prove this. So if we change the identity of the father now to be white, so his leaves are mostly white. Now, some of you may be saying, how does a plant survive without any functioning chloroplasts? The answer is simple. Even though the leaves are white, they still contain enough chloroplasts that are functional to keep the plant alive. Maybe not to the same size as a wild type plant that's got green leaves, but still these 4 o'clock plants are alive. So in this case, you breed a green female plant with a white. And the answer is all the offspring are still green, because the father does not contribute anything to the F1 in terms of the chloroplasts. All right, so let's now look at a different situation. So let's get rid of some of these things. There we go. And we'll go back to here. And now we'll change the mother to a different color. So let's make the mother white now. So all she has to put into her eggs is the mutant chloroplast. And let's say the father is green. Therefore, the father is blocked because he's not allowed to contribute any chloroplast DNA. And therefore, all the babies should be white. Even if the father was white, it would make no difference. Even if the father was variegated, it would make no difference, because nothing can get through the pollen into the offspring. So that leaves us with one more choice, right? Let's change the mother to the third type. So we had a green mother, we now have a white mother. Let's go for a variegated mother. And this is where things get a bit interesting. So let's erase a few things. Let's erase this. Let's erase all this. And then we're in a position to erase her phenotype. So now let's change the mother to variegated. What would be the outcome for the child? Now this is interesting. To be variegated, that means her body contains both green wild type chloroplasts and white mutant chloroplasts. And the question is, which ones were present inside the egg when the egg was made from her other tissue? So if that whole branch of her body, the mother, was made of just green chloroplasts, then the chances are that the egg that was formed from this tissue would only have green chloroplasts. And therefore, even though her body as a whole is variegated, she may only pass on green. So one possibility, let's call it A, is that all her offspring will be green. Now reverse the situation. Maybe the flower now evolved. The flower developed on the end of a branch that was purely white. So even though the whole plant is variegated, maybe that particular region was just white. So it only contained white chloroplasts. Then in that situation, her offspring may only inherit the mutant chloroplasts. And the third situation is where the region of the egg had both types of chloroplasts. So the tissue that gave rise to the egg, that was able to pass in through its cytoplasm both the mutant white chloroplast genomes and the green wild type. And in that case, we have a third option. Now the baby will inherit both types of chloroplasts and it will be variegated, right? So some parts of her will have green, the baby that is, and some parts of her will have white. So she has both types. You can have tissue with a mixture of either pure green, pure white, or a combination of white and green chloroplasts. But everything goes back to the mother. The father does not give anything. So that's what you have to remember. The best way to learn this is to change the parents, especially the mother, and see which outcome you can predict, and then compare that to the answer. So same thing as mitochondria, but a bit more complex. Can we give you a biochemical explanation? Well, we just did in a form, but the biggest thing you have to understand is that if this is the tissue that will develop into the egg cell with its haploid version of the chromosomes, so this is the egg, and this is the tissue that will develop the egg, the ovary, then it depends on what's inside and how they get segregated when the cell divides to form this particular cell. If all the chloroplasts on this side are green, then the egg will have nothing but green. But if you have also white, then the egg will also be white. So it'll have both types. So you can either be all green in the egg, you can be all white, or you can be a mixed. The most common is going to be mixed, but not always. It depends on which part of the plant the flower is developed from. So I'll leave you with that to contemplate. Let's move on to a very quick understanding of imprinting. Now, before we can look at imprinting, we need to understand epigenetics. So epigenetics is simply taking this word here, which says above the genome, and not looking at changes to the DNA bases. So we're familiar with that. ACGT would be the basis of DNA. They're not changed. The information that's inherited in terms of the basis is exactly as we would predict with Mendelian genetics, right? What's going on here is the compactation of the chromosome, the regulation of the genes in terms of compact versus loose is regulated. So what that means is this. Suppose you have a region of DNA that contains a few genes. If you take that DNA and you make it very tight by compacting it, by twisting it around and round, then these are the 3 genes. The genes are so tightly packed that the enzymes that are supposed to activate the genes, they can't get in there. It's like taking your hair and turning it into a knot. There's no way that the comb can get in there, right? Where if it's loose like this, the comb can go through quite easily. So this will be turned, this region of the chromosome will be off, and this region of the chromosome under this circumstance will be on. So that's what epigenetics does. It changes the compactation of the chromosome. It makes some regions tighter, other regions open up and loose. And this can be done at various levels. It can be done at the gene level, where one gene is shut down and it doesn't affect the neighbors. It can be done at a large chromosomal level, where hundreds of genes are involved in that compactation event. Indeed, it can be done for an entire chromosome, where the entire chromosome is shut down. And if you take a few seconds to think back in the video lecture on bar bodies and X chromosomes, you will notice that's exactly what happened there. So we have 2 X chromosomes inside the female. One of them will be shut down into a bar body. And bar body is an example of that. So here an entire chromosome with over 1,000 genes is all shut down and made into a compact body. And there are examples of a whole genomes being shut down, a whole chromosome sets. So you can inherit 3 sets of chromosomes where one whole set is shut down. Every single chromosome in that set is condensed through epigenetic manipulation. How does epigenetics work? And there are a number of different ways by which this system works. Let's just go through and look at one. Let me use a different color. So here's your DNA. And here's a region of a gene, is another gene. And let's compare that to another identical piece of DNA on another chromosome. So what happens is the C bases, Cs, are randomly distributed up and down the DNA, right? Not so randomly that it doesn't make information sense, but you know, in terms of their positioning, as far as we look from the outside, they're in certain places, there's no pattern. So what the system does, it has a bunch of enzymes, and these enzymes can add chemical tags to this DNA backbone. Not only that, it can also add chemical tags to the proteins that are taking care of the DNA. So as you know, chromatin is the name for DNA plus protein. So this is chromatin. And working alongside the chromatin are these enzymes that can add and remove these chemical tags, right? So if you put a chemical tag on this, like a methane group, methane, carbon plus 3 hydrogens. So this methyl group can be attached, and the more methyl groups you attach, the more likely that the chromosome will become compact. So the chromosome becomes compact when it's methylated. And if this chromosome is not methylated at those Cs, it's left alone, then it stays open. Therefore, this chromosome and these 2 genes, this gene will be on, this gene will be on, and because this gene has not been methylated, it'll stay on, whereas this gene, which is methylated in various places, will be off. And this pattern can be passed from one generation to the next. And it takes a few generations to reset. Let me give you a great example. Recently, we found that your diet at this time, your lifestyle in terms of your diet, will influence this pattern of modification of your DNA, and it will be passed on to your children. So if you have a good diet, they have a good health outcome. You have a bad diet in this lifetime, then when you have children, you'll pass that badness onto them. And it'll take them a couple of generations before they can wipe the slate clean by eating healthy. So methylation is one form of epigenetic modification. What we're going to do now is look at imprinting in a classic case, right? Let's consider this with respect to an example. The classic example in genetics is called IGF. IGF. And there's many versions of IGF. We're going to look at IGF2, this particular gene. I stands for insulin, and G and F stand for growth factor, right? So this is going to be insulin-dependent growth factor. As you know, insulin is very important in regulating blood sugar, but this is a growth factor. So this is a different type of protein than insulin. And this one is involved in growth. So if you have a defect in this particular protein, then you're not going to grow to the same potential, and then you'll be dwarf. Okay? It's a recessive condition. So the mutant version is recessive and is indicated by a minus sign. So let's put down both alleles. Okay? So this is the wild type, and this is going to be the mutant version, right? We use minus. And we're going to use the same example as most textbooks, which is to look at mice. And remember, the goal is to help you understand imprinting. If we have a male mouse and we breed it with a female mouse, and both mice are wild type for the condition, then of course their babies will be normal. And the entire litter will grow up to be full size. So that's obvious. So let's change the settings slightly, and let's change the appearance so we understand this. So we'll keep the same colors. This time, let's make the father heterozygous and the mother too. So his type of sperm will be positive and negative, and her type of eggs will be positive and negative. The question is, what do you expect to get? And the answer is, it depends on the father's sperm, and only on the father's sperm. So in this case, it's irrelevant what the mother is. It's irrelevant. That's what the experiments revealed. So let's give you a simpler example of this, because this is the ultimate example. We'll come back and look at that later, right? So here's the father, and here's the mother. So if the father is mutant, and the mother is wild type, and all her eggs are wild type, and all his sperm are mutant, and then they fertilize, then what will be the babies in the F1? Will they be normal, or will they be dwarf? Those are the phenotypes. And remember, the mother has no impact in this case. The gene in the mother is turned off. And we'll go into why in a second. But the father's genes, the one gene that's passed through the sperm, is on. So in this case, the only thing the babies will receive is a mutant form of IGF. So in this case, all the babies will be dwarf, 100%. So let's change the situation and do a reciprocal cross, right? Let's change the parents around. So let's just use this. Let's get rid of this, this, and this, and that, right? And now let's put that situation in. Let's change the color slightly. So this time, he produces normal sperm, and she produces mutant eggs. Upon fertilization, she doesn't matter in this case. And therefore, all the F1 are going to be normal. Because it's only the father's sperm that have the active form of the gene, unmethylated. The mother's egg, they all contain methylated regions corresponding to this gene, right? So the region around this gene, including the gene in the mother doing egg formation, is always methylated. Doesn't matter what condition that chromosome was before. When it arrives into the ovaries, it's methylated. Let's consider one last case, just to get you used to this. So let's go for a heterozygous outcome, just like before. So let's get rid of the appearance. And we get rid of the eggs. Now let's see what would happen in a double heterozygous mating. Okay, so here we go. So the father is going to be heterozygous, and the mother is going to be heterozygous. So she forms two types of eggs, and he forms two types of sperm. Remember, it's irrelevant what the eggs are. So it doesn't matter whether they carry positive or negative. In terms of the gene itself, that region of the gene is shut down. In the case of the father, we have two types of sperm. If the babies are born of the top sperm, they'll be normal, right? If they're born from the bottom sperm, they'll be dwarf. To understand this at the biochemical level is very important. So let's go and have a look at that. So let's get rid of everything on this page, and then let's move our migration to look at what's going on during gametogenesis, right? So let's have a male over here. Let's have a female down here. Okay, so this is the parent, and this is gametes, and then this is the fertilization, right? F1. So the parent can be anything you like, right? So the parent could be plus plus male. The parent could be plus minus male. The parent could be a minus minus male. So in this case, the father is dwarf. Here the father is heterozygous normal. Here the father is heterozygous normal size. It doesn't matter what the father is with respect to the epigenetic behavior. What happens is this. Whatever the chromosomes, they enter into spermatogenesis. And the actual alleles are unaffected. Unaffected. Now, you may be thinking, well, what about the ones that came in from the mother? So one of these two chromosomes must have come from the mother, right? So suppose this one came from the mother and this was compact. And suppose out of these two, this one came in from the mother, it's just compact. Suppose out of these two, this came in from the mother, which is already compact. It doesn't matter. When it goes through gametogenesis, the tags are removed. So remove all tags. So all the methyl groups are removed from this gene. And therefore, it doesn't matter what we have next, everything is untagged. So whatever goes into the gametes, the sperm, in this case on the top example, all the sperm will be carrying the wild type allele active. In the case of the heterozygous, all the sperm will be carrying active forms of those two alleles, right? And then on the bottom, then all the alleles there will be active. They may be mutant, but they're still available to be expressed, okay? So all the sperm are now fine. In the case of the mother, we again have the same situation. So maybe this chromosome came in from the father, and this one was compact, came in from its mother, right? So in this case, what happens in spermatogenesis, what happens in oogenesis, right, is that tags are put on. So all alleles are tagged. Therefore, none of them will be active. Doesn't matter what its genotype is, right? So in this case, you have all plus eggs, but they are tagged, they're compact. In this middle case, you have two types of eggs, and they're compact, both. And on the bottom, you only get one type of egg, and that's compact. So whenever you get fertilization, whatever the father happens to give the baby, that's the genotype that will then be affecting and producing the phenotype. The mother has no say in that information. So if you get a minus sperm, then the baby will have a IGF-2 that's broken. It won't be able to produce the protein, therefore the baby will be dwarf. If it happens to be born from a positive sperm, where the IGF-2 is normal, then it'll be a normal mouse, right? And you can see that's going to affect the ratio of which these progeny are born. So if there's two types of sperm, there'll be 50-50. 50% will be normal, 50% will be dwarf. If there's only one type of sperm, if it's a positive sperm, all the babies will be normal. If it's a negative sperm, all the babies will be dwarf. So think about that, please. All right, let's go on to the last one then. This is my favorite, like I said earlier, maternal versus paternal. Which one supplies the biochemistry for beginning life, right? So this is all to do with how the mother's ovary, where the egg is being both generated through meiosis, and what happens inside the mother's uterus when the egg is fertilized, right? In most cases, the sperm doesn't have a say yet in what happens to the biochemistry. So let me do the biochemistry first. So here is the developing egg, and it's surrounded by the mother's normal cells, right? So these cells are called nurse cells, because they nurse the oocyte. And whereas this thing is haploid, these cells are diploid, right? So we'll just put diploid for some of them. Depending on the biochemistry of these cells and their DNA and their nucleus, they will then leach chemistry onto and into the egg, therefore affecting what the egg can do in the next number of hours or days, okay? So the example that we're going to use is going to be referring to a particular gene that we call, in this case, we're going to use snails, right? Because the sea snail is the exact freshwater snail, sorry, is the example that everybody uses, right? So pretend that we're looking inside the ovary of a female, and there's two ways that the shell can spin, just like, you know, when you flush the toilet, the water spins in a particular direction as it leaves the toilet. That depends on whether you're in the northern hemisphere or the southern hemisphere. In the northern hemisphere, it spins one way. In the southern hemisphere, it spins the opposite way. So the same thing can happen with respect to the shell coiling of these snails. It can coil one way, clockwise, or it can coil the other way, anti-clockwise. So when it's right-handed, it's going to coil this way, right? And the thumb tells you which way it's going, upwards, and it'll coil in that direction, anti-clockwise. And then you have the other way, which is the other hand, and that coils the other way, which is clockwise, right? The dominant one is this. It coils to the right. And that's controlled by a single gene that has two alleles. So let's represent dominant with D. So you have two genes, sorry, you have one gene with two alleles. So you could be heterozygous, homozygous dominant, or homozygous recessive, right? So let's put them down. So this refers to the mother's cells. So this nurse cell up here could be either one of these combinations, right? So if it's DD, then it's going to produce a chemical and leach that chemical into the developing egg. And when that egg gets fertilized, it's going to then have an impact on which way the animal develops, whether it's going to have a right-handed or left-handed shell. So remember, big D equals right-handed, right? So in this case, all the cells will have big D, like this. And therefore, they're all leaching in a chemical into the developing egg, which upon fertilization will start mitosis, get the right word, will start mitosis by forming a right-handed spiral to the body. So that's what happens here, right? So it doesn't matter what the baby is. The genotype of the baby makes no difference. That's what we mean by maternal effects. The mother's genotype decides. The baby doesn't have a chance yet to influence its own outcome. So the baby could be whatever. It makes no difference. So ignore the baby. So the baby could be big D, small d, or small d, small d. That makes no difference. It's the mother's biochemistry that's important here. So let's put down all the examples. So let's get rid of this and let's put it into 3 columns, right? Okay. So here the mother's genotype is homozygous dominant. Here the mother's genotype is heterozygous. And here the mother's genotype is homozygous recessive. So in this case, we just put a couple of nurse cells in, right? So here the nurse cells are big D, and they will release a chemical that will cause that to have a right-handed shell, right? Here, the nurse cells are heterozygous, but still, because big D is dominant, they will release a chemical that will cause this to become right-handed. However, here, the nurse cells are homozygous recessive. So they're completely without function. So they don't release anything into the mother's developing egg cell, and therefore it develops into a left hand. It doesn't matter what the father is, because by the time the sperm arrive, it doesn't have enough time to change the biochemistry of the cytoplasm of the egg cell. Remember, this is affecting the cytoplasm of the egg cell. So this is sharing biochemistry across from the nurse cells into the oocyte. So this is called the maternal effect. Therefore, the genotype of the mother is the most important thing. What you have to do is be able to predict what would happen to a baby that was of this type if it was born inside a mother of this type. What would happen to that baby if it was breeding with another snail? What type of offspring would the F2 be? And then can you predict the F3? So once you reach that understanding level, then you have succeeded in understanding maternal effect genes, right? And in most cases, if not all, the mother's genotype is responsible for dictating the phenotype of the baby at this particular stage. Thank you so much, and we see you next time.